Come on, get your lab coats and safety goggles on. We are heading over to the biology lab because it's time for our favorite type of lesson, and that is the practical. All that you have learned so far regarding the stomata, we will now be seeing that knowledge come to life. That is so exciting, isn't it? We will be performing an experiment to investigate the effect of light on gases exchange from a leaf, both at night and during daytime. And this is where the stomata come in. We already know that the stomata are the microscopic pores that are present on leaf surfaces, mostly on the lower epidermis. Not only do they provide a passage for the gases to pass through, but these efficient little multitaskers allow water vapor movement as well. Now, we already talked about how leaves only photosynthesize during the day, but respire all the time, right? So this means that the workload of the stomata changes as time progresses from day to night. But the good news is that you can easily observe this workload directly. Now, are you wondering how? Well, let's begin the practical. Go ahead and grab a few petri dishes, some distilled water, some glass slides, cover slips, and the stunning blue stain called methylene blue. Oh, and of course, a light microscope. Now we are ready to begin with the experiment. We are going to take a fresh thick leaf here and peel off a very thin layer from its lower surface. We will carefully place this in a petri dish that is full of water so that the layer does not curl up. Now we are going to cut up a fine square piece from this epidermis and place it on the glass slide. We will then carefully add a few drops of water first, and after that a few drops of methylene blue. You will soon get to know why this dye is needed. Now here comes the tricky part. Slowly and very carefully place the cover slip onto the slide, avoiding any air bubbles from forming. So with this, we are ready to observe the preparation on our light microscope. First, we are going to set the microscope on low power. Next, we are going to view the slide. And then we are going to move towards the high power setting. We will repeat this procedure twice. First, during the daytime, and then during the night. Each time at both day and night, set your microscope at lower power and take a nice look. Count the total number of stomata you can see. Then take an even closer look and identify the ones that are open. The open stomatal pores are the ones that are actually active, meaning they are undergoing gases exchange. Now make an observation column just like this. Use it to compare the total number of active stomata at both times. So, can you tell me when the stomata are more active? Okay, that must be a piece of cake for you now. Oh, and how many stomata were you able to count? Wait, our work is not done yet. It's time to take an even closer look. Go ahead now and increase the setting to a higher power and have a look at the structure of the guard cells. Ponder over how these bean-shaped guard cells actually function, allowing the opening and closing of the stomata, thus controlling gas exchange. Draw these structures out, label this diagram, and with arrows indicate the movement of gases through it.